Dear students, I am Bharat Kashyap, lecturer Biology at GSSS Majwad, District Mandi. Today I am going to teach you the fifth chapter of Biology, Principles of Inheritance and Variation, and today's topic is Mutation and Pedigree Analysis. In this video, we will study the following subtopics. To study what are mutations, to study different types of mutations, method used to study human genetics, to study the dominant and recessive pedigree charts. Mutations. The term mutation is given by Dutch botanist Hugo de Vries in 1903. He observed variations or mutations in plants of Oenothera lamarckiana. Its common name is evening primrose. Mutations are changes those occur in DNA sequences which alters the genotype and phenotype of an organism. Mutations are sudden, stable, discontinuous, and inheritable variations due to change in genotype of organisms. They are caused by substances called mutagens. Now, types of mutations. Mutations are mainly of two types. First one are point or gene mutations. The other are chromosomal mutations. The point or gene mutations occurs when there is a change in one or more gene. They are of following types: substitution, insertion, deletion, and frame shift. Type of point mutations. The other one are chromosomal mutations. The chromosomal mutations are of further of two types. First one are morphological and other are numerical. The morphological chromosomal mutations are those where structure of chromosome alters. They are of following types: deletion, deficiency, duplication, inversion, and translocation. Whereas the numerical chromosomal mutations are those where chromosome number changes. They are of further two types. First ones are aneuploidy. Here the chromosome number is not exact multiple of basic haploid number. There are following types: monosomy, nullisomy, trisomy, and tetrasomy. Whereas in euploidy, the chromosome number is exact multiple of basic haploid number. They are of following types: haploidy, diploidy, triploidy, tetraploidy, pentaploidy, hexaploidy, and so on. Point mutations. They arise due to a change in a single nitrogen base pair of DNA. Look at the diagram here. This is single base pair AT. It is replaced by TA. It results in change of amino acid and protein. Look here. It results in change of amino acid. Instead of glutamic acid, valine is formed. Hence, instead of normal protein, mutant protein is formed. For example, sickle cell anemia, where one gene hemoglobin A is mutated with hemoglobin S. It replaces glutamic acid in chain of hemoglobin A with valine amino acid. Thus, RBC becomes deficient in oxygen, and due to the tension of deficiency of oxygen, they become sickle shaped. And in this diagram, you see they block the blood capillaries and make the organs deficient in oxygen. That's why we call it sickle cell anemia. Types of point mutations. First one is substitution. When one nitrogen base pair is replaced with another, this is called substitution type of point mutation. Look at the diagram here. This is the cytosine nitrogen base. It is replaced with thymine nitrogen base. So this is the substitution type of point mutation. Next is insertion. That means when an extra nitrogen base is added to DNA. So in this diagram, uracil is added here. So UAC changes into UUA, and GGG nitrogen base triplet changes into CGG, and at the same time the amino acid also changes. This is the insertion, effect of insertion. And now the deletion. When a single nitrogen base is deleted, look at the picture here. This is the cytosine is deleted. Now UAG, this is the G comes from this code, triplet code to here, and UAG is the stop signal, stop codon, hence no further formation of amino acid or protein take place. So these are the different types of point mutations. Now frame shift mutations. And during frame shift type of gene mutation, addition or deletion of single nitrogenous base takes place. None of the codon remain in the same position, and reading of genetic code is shifted laterally either in forward or backward direction. You can see here, this is the base triplets in DNA, TAC, CAT, TAG, ATT. Now, this is the insertion. As we have already studied, one nitrogen base is added, cytosine is added at this position. So it becomes TACCCA, and all the frame, the different types of triplets changes beyond this. CAT now becomes CCA, TAG now becomes TTA, ATT becomes GAT, and one thymine is left outside. Similarly, when one nitrogen base thymine is deleted from this chain, so at this position TACCAT, now thymine, this T is deleted, it becomes now AGA, AGA and TT. So as the genetic code triplet changes, the amino acid as well as protein also changes. So this is the frame shift mutation. Now chromosomal mutations, and first are morphological chromosomal mutations. Its first type is deletion. When loss of a segment or part of chromosome take place, it is called deletion. You can look here. This is the chromosome. The D segment is missing here. So this is deletion. Now, duplication. It is a type of chromosomal mutation that occurs when a section of a chromosome is repeated. Again, it is quite clear in this diagram. BC is the segment. It is now repeated. BC, BC. So this is the duplication. Next is inversion. In inversion, a segment of a chromosome is reversed end to end. This is BCD. It is reversed DCB. So this is inversion type of morphological chromosomal mutation. Now, translocation. When a part of one chromosome attaches to another non-homologous chromosome. So it is of two types. Simple is one, when one chromosome transfers a part to another chromosome, and other is a reciprocal type of translocation, when both the chromosomes exchange their parts with one another. Now look at the diagram here. This is showing reciprocal translocation when A, B, C, D, E, F. This is one chromosome, and M, N, O, P, Q, R. This is another type of chromosome, non-homologous one, because its genes are different. So when we are saying reciprocal translocation, that means both the chromosome exchange their parts with one another. So ABC, it comes to this chromosome, whereas MN goes to this chromosome. So this is a reciprocal type of translocation. Numerical chromosomal mutations. First of its type is aneuploidy. Look at the diagram here. Aneuploidy, it can be defined as a condition in which a person has one or a few chromosomes above or below the normal chromosome number. Its first type is monosomy. It occurs due to loss of one chromosome. This is the normal chromosome complement, and monosomy, you are seeing here one chromosome is missing. This is monosomy, and its example is Turner's syndrome, which occur due to loss of one X chromosome. Next type is nullisomy. In this type, a pair of homologous chromosome is missing. This is the nullisomy, when a pair is missing. Then trisomy, when three copies of chromosomes are present. This is trisomy. For example, Down's syndrome, which occur due to trisomy of 21st chromosomes in human beings. Next is tetrasomy. This is tetrasomy. It is due to presence of 4 copies instead of normal 2 of a particular chromosome. So this is tetrasomy type of mutation. Next is euploidy. Look at the diagram. In euploidy, an organism acquires an additional set of chromosomes which is over and above the diploid number. Depending upon the number of chromosomes sets, they are of following types. First are monoploidy, that is, they have a single set of unpaired chromosomes. They are also known as haploids. Then there is diploidy. when two sets of chromosomes are present. This is the normal diploid number found in higher organisms in their cells. Then polyploidy, when more than two sets of chromosomes are present. This is known as polyploidy condition, and polyploidy is of following types depending upon number of additional sets. First are triploid. They have three sets of chromosomes. Then tetraploid. They have four sets of chromosomes. Pentaploids. They have five additional set of chromosomes. Similarly, hexaploids. They have six sets of chromosomes. So this is euploidy. Generally, it is found in plants. Pedigree analysis. After the rediscovery of Mendel's principles, the inheritance of traits in humans began. The controlled crosses made by Mendel on plants cannot be performed in human beings due to the following reasons: long time between generations, small families and a few offsprings, impossible to make experimental marriages. For study of human genetics, a record of the inheritance of a trait in several generations of a family is kept and called pedigree analysis. It is a strong tool to trace the inheritance of a specific trait, abnormality, or disease in a family tree. Let us study the various symbols which are used in human pedigree analysis. Male members are shown by squares. Female members are shown by circles, and this symbol is used to represent unspecified sex individuals, and if these symbols are darkened, they represent affected individuals, and there is a horizontal line between male female, it represents marriage between them or mating, and if it is double line, it means mating between close relatives, also known as consanguineous mating, and it leads to recessive syndromes or diseases. Then this is showing the first generation parents, and vertical line showing the second generation. These are the children in order of birth from left to right, and in this case the parents with male child affected with the disease, whereas this is showing that 5 unaffected offsprings. Let us study some common pedigree. First is autosomal dominant. In autosomal dominant pedigree, the trait tends to appear in every generation of the pedigree, and that the affected fathers and mothers transmit the trait to both sons and daughters. For example, myotonic dystrophy. Then we have autosomal recessive. In autosomal recessive, it is one of several ways that a trait, disorder, or disease can be passed down through families. An autosomal recessive disorder means two copies of an abnormal gene that must be present in order for the disease or trait to develop. Its example is sickle cell anemia. The person from whom case history of a pedigree starts is called proband. If the proband is male, it is called propositee. If the proband is female, it is called proposita. Pedigree analysis is useful in sexually reproducing organisms where the number of offspring is small. The pedigree provide information to the would-be couples regarding the danger of certain genetic defect which might run in their family. With this, we come to close of today's lecture. I hope you must have understood it properly. Thanks for watching.